Welcome. I'm Clary Simpson, CEO of the CMT Research Foundation. And on behalf of the Foundation's Board of Directors, the staff, and the Scientific Advisory Board, we welcome you to the third annual CMT Global Research Convention, and only our second in person. The program for our 2 days together will be challenging and inspiring, I promise you. We have a packed agenda, so for the speakers, please don't mind if you see one of us waving this at you in the background, all right? Take no offense, but we have lots and lots to discuss. And we've also built in plenty of time to continue conversations during lunch and in the evenings. We promise that too. As the parent of a CMT patient, I'm honored to work to support your research to develop treatments for all CMT types. We are so pleased to see you here this morning, eager to participate in the sessions, engage in discussions, connect with colleagues you know, and make new acquaintances. We all stand together with our diverse experiences and different skill sets, bound by a shared determination to overcome the challenges that CMT presents. So it's really hard for me to believe that the Research Foundation is celebrating its 5th birthday this month. In this short time, we've funded 20 projects, and of those, 5 already have preclinical candidates for CMT1A. The foundation funding has also gone to CMT1B projects, 2A, 1X, 4B1, and 2E. And there's more to come because we want to cure all types of CMT. Now, for the elephant in the room, I know that many of you, especially the patients here, really want to ask questions and talk about DTX Novartis. But please keep in mind that it's early days yet in the acquisition. And as many of you know, these integrations take a good deal of time and quite a bit of patience. We're lucky to have Artie Suko, who I can't see because I have the wrong glasses, founder of DTX, and Brian Lafitte, who is the current Chief Scientific Officer of DTX Novartis, in the room with us today. And not only do we want to congratulate you, but we want to thank you for all of your hard work and for creating so much momentum for everyone involved in CMT research. So thank you and congratulations. I don't think they're allowed to answer too many direct questions, however. But I also want to reassure you that CMT Research Foundation has been in close contact with the team at Novartis. That team is led by one of the most esteemed CMT experts that many of you know, Robert Balow. And we're really encouraged as they're working very hard to create a plan for the continued development of DTX 1252, which we hope will be the first effective treatment for CMT1A. When that plan is clear, we'll let you know. You can be certain that we will share the information about clinical development and trials as soon as it's available to us. So we have a much larger group in the room this year, for which I'm very, very grateful. And I hope that you're going to ask a lot of questions. We will have roving mics for any of those that are in the room. I would ask you to wait for the microphone to ask your question so that our virtual audience can hear as well. And I'd like to welcome our virtual audience and let you know that you can use the Q&A function, and Dr. Robert Pryor over here to my right will make sure that your questions are answered. Rob is with us again this year, and we're very happy for that. He's a postdoc researcher at the University of Bonn, working in Professor Volker Buskamp's laboratory. He's deeply involved in researching neurodegenerative diseases, specifically type 1A and X-linked type 1. Thank you, Rob, for your help. You really are very adept at this. And I know as a person living with CMT and a scientist who studies it, this gathering is especially important to you. So please know that you are a dear friend to us. Now, before I hand things over to our scientific programming committee chair, I'd like to thank our generous sponsors for their support of the convention. You can see them listed over there. Among them are Nanite, Augustine Therapeutics, Samsara Therapeutics, Armatus Bio, Horizon, Mitochondria Emotion, Takeda, and DTX Novartis. Thank you. Now, I think all of you know Dr. Charles Abrams, head of the Neuromuscular Division at the University of Illinois Chicago Medical School and renowned CMTX researcher. Charles is also a member of the CMTRF's Scientific Advisory Board and chair of this year's Convention Scientific Programming Committee. Thank you, Charles, for all your work, and you have really assembled a remarkable program for the next 2 days.
It just occurred to me, it depends where you put the comma between X and researcher. So welcome, everyone. As the chair of the Committee for the Scientific Program, and speaking on behalf of the entire committee, I extend to each of you a warm welcome to the 3rd Annual Global CMT Convention. The convention is a cornerstone event for advancing knowledge and collaboration in Charcot-Marie-Tooth disease. This year's program has been carefully formulated around some of the most pressing topics in the CMT space. We hope that you will engage in these discussions and share your expertise and contribute your insights. This convention is really a unique opportunity for us to learn from and connect with one another and to accelerate progress in finding a cure for all types of CMT, which is really what we're all here for. I would like to thank the other members of the Scientific Programming Committee, Tina Irv of the U.S. National Center for Advancing Translational Sciences at the NIH, Paul August of Revere Therapeutics, Glenn Nichols, also at NIH in the National Institute of Neurologic Disorders and Stroke, Amanda Hayded-Phillips at Sarepta Therapeutics, and Afrouz Rosh-Nonajad at Nationwide Children's Hospital. I would also like to thank you, the attendees, for being here, for your dedication to this vital cause, and for your commitment to making a meaningful impact in the world of CMT research. It's now my pleasure to introduce our Chief Scientific Officer, Edrich Havalosa, who joined the foundation from the Muscular Dystrophy Association early this year, and she will kick off day 1 of the Global CMT Research Convention. Edrich, oh, there you are, right?
Thank you, Charles, for the introduction. Wow, this is so exciting, seeing everyone, some familiar faces and new ones as well. So thank you for being here and welcome. Welcome, everyone. We're really excited to have you join us in person and also virtually. I hope you all had your caffeine intake today because we do have a lot going on and we have a full program. It's very exciting, so I don't want you to miss anything. So if you haven't had your coffee, or I think, well, hopefully they'll come in here in the room soon. It really is an honor to be here with you all today. Before we start the sessions today, I would like to take this moment to share my experience so far working with the people at the CMT Research Foundation. Since day 1, I have witnessed the efforts from my colleagues to keep the momentum going to really find the cure and treatments and cures for CMT, which is the mission of the foundation. Our foundation may not be the largest, but our determination and dedication run deep. The work the team has accomplished had so much commitment, and this is really I can actually see it from my team. Every day there's so much momentum, and it's so infectious, right? And it affects the work that we're doing. And what we're really trying to do is to help those affected by CMT, and that commitment is what makes this dream the dream team. As we come together for this convention, I can't help but feel a tremendous sense of pride to be part of this amazing journey. Today's schedule is packed with valuable insights, meaningful discussions, and sharing the cutting-edge research that's set to advance our understanding of CMT disease. We'll be listening to some of the most brilliant minds in the room and really throughout in the field of CMT, diving into the latest scientific breakthroughs and engaging in strategic conversations. This convention is a chance for us to untangle complexities of CMT, and as importantly, to also encourage and foster collaboration among all of us here. It's not just about gathering knowledge, it's about generating new ideas. and forming partnerships and fueling that flame of innovation. Together, we can really make meaningful strides in CMT research. So I know we're already running behind time, so I don't want to take too much here and get in trouble for taking up so much stage time here. Well, we have a very, very busy agenda, so I'm going to just wrap things up here. In closing, I really sincerely want to thank you all for being here today. Your presence demonstrates your dedication to this cause, and together we can really find treatments and cure for CMT patients and their families, and they can look forward to a brighter quality of life. Again, thank you for joining us on this important scientific journey. And I hope you really do enjoy our program today. Our scientific programming committee have worked really hard to put this together along with the team at the CMT Research Foundation. So moving on to our first session of the day, I would like to invite Patrick Levny, who is the co-founder and chairman of the board of directors of the CMTRF, who will introduce our keynote speaker. Pat personally has CMT and has been a strong and outspoken advocate for CMT patients for more than 20 years. So Pat, come on up.
Well, good morning, everyone. Before I start, I want to thank Artie for keeping me out till 12:30 last night. So I'm going to keep my my comments brief. Welcome, everyone. I'm humbled by the brain power in this room, all focused on our mission, which is to end CMT, find the first treatment / cure. And I couldn't be more proud to be part of this organization and the hard work that's been put in. I might tell you there are two people that are near and dear to my heart and the organization's heart that when it wasn't popular, jumped in with both feet to help Susan and I 5 years ago craft what you're seeing now as a result of that work. One is Paul August, and the other is Charlotte Sumner. It wasn't popular back then, but they said, you know what? We like the mission statement. We like the mission. We're in. And I might add that I've known Paul, and he was the first one that got pharma involved with Sanofi Genzyme, which I'm grateful for as well. So Charlotte, you hold a special special place in our hearts at the CMTRF. Susan, myself, Paul, and Charlotte, when we came to you to shape what we're going to focus on as far as the gaps, as far as what we needed to solve for to get to that first treatment, was in DC. You set the agenda for our focus, whether that's delivery systems and the other three pillars of what we were trying to accomplish. And guess what? You were dead on. And the result of that is what Clary had mentioned to you, 5 clinical candidates in very short time, DTX. There's a couple more announcements that are coming over the next couple of weeks. So if you dare to challenge, if you dare to do things different, it's amazing what can happen for the positive on the mission. So Charlotte Sumner, I'm going to do a brief introduction. We tapped Charlotte on the heels of her success in SMA. Charlotte's a professor of neurology and neuroscience at Johns Hopkins University School of Medicine. She's really focused on individuals with inherited neuromuscular disorders of peripheral nerves and motor neurons, including SMA or spinal muscular atrophy and CMT. She co-directs the Johns Hopkins Muscular Dystrophy MDA Care Center, the Spinal Muscular Atrophy Clinic, and the Charcot-Marie-Tooth Clinic as well. These clinics deliver multidisciplinary clinical care, engage in international natural history studies, and provide cutting-edge therapeutics. Her research focused mainly on developing treatments for degenerative disorders of motor neurons and peripheral nerves. Her lab uses human tissues and iPSC cell lines, mouse models, cultured cells to characterize these disease mechanisms. One of the tenets of our organization is we want to learn from winners. Charlotte is a winner in the SMA field, is a winner in a big part of the CMT community. We go to the cystic fibrosis for advice, and we shape that towards a successful mission that we have here. So without further ado, I can give you about 20 pages of CV and the writings and everything else that Charlotte has done. Here's Charlotte Sumner.
Okay, so I'm going to have to go even faster than I was intending. But thank you so much, Pat, for the introduction. And it is so impressive what CMTRF has achieved in just 5 years. It's really fantastic. And I hesitate to give this presentation because I'm really preaching to the choir here. I think we all know that no one person ever achieves therapeutics development in a disease. It involves a whole bunch of people, whole bunch of stakeholders. And so what I was asked to talk about today is really the power of that collaborative effort. So when I think about neurology and neurotherapeutics, and certainly when I was training, you know, we were probably somewhere down here. Here's neurology and here's the therapeutics. So, you know, very few therapeutics available for these devastating diseases. But I think something's changing. I'm really hopeful something is changing and that we're actually, we're actually on this trajectory where the slope of therapeutics development is changed and is changing quite rapidly. I'm very hopeful that very soon we're going to be up here. And part of the reason that that's happening is that just this tremendous step forward that's been made in gene targeting therapeutics, which we can now apply to multiple neurogenetic diseases. And this stat from the FDA that 10 to 12 cell or gene therapy approvals are expected each year going forward in the near future is kind of stunning to think about. So I like this quote: if you want to go fast, go alone. If you want to go far, go together. Of course, we'd like to go fast and far, but anyway, I think you get the point. This effort to develop treatments for disease requires so many different people. And one of the biggest challenges is trying to get people to do this together, to work together. And part of why that's challenging is because sometimes we speak sort of different languages. That can certainly be the case between basic scientists and clinicians, academia and industry, and so on. But it's absolutely essential that these conversations are happening and that there are real deep efforts for all of us to be working together. And this sort of scheme lists some of the stakeholders that are needed in this effort, but I have patients and families and disease foundations here very purposely at the top of this interacting matrix, because I think they, particularly in the rare disease space, play such an important and fundamental role in making sure this jigsaw puzzle is put together and put together effectively. They are the ones who can create forums like this, where we can all come together and actually have conversations about what needs to be done to move the field forward. They can reach out individually to all the other different stakeholders and connect key people that allow us to make steps forward. So I really, I think that is an absolutely essential role for these folks. So I work on SMA and CMT, and SMA, as everyone knows in this room, has experienced some success, which is fantastic. That may mostly have been luck. I don't know, but I firmly believe that the that the community of stakeholders in this space actually was a major reason that this field succeeded. This is just kind of a timeline of therapeutic success in SMA. You know, the disease was described pathologically in the 1890s. The genetic cause was identified in 1995. And once that finding was made, it actually, things moved pretty quickly. I mean, of course, we always want things to move faster, but, you know, 5 years later, we had the first animal models of the disease. NIH actually had a special targeted program devoted to SMA, saying, you know, this is the disease that's closest to therapeutics development, and invested money. And of course, there were efforts, parallel efforts, both in cell models, mouse models, with ongoing efforts in the clinical space to develop clinical motor functional outcome measures that were relevant for CMT patients. And, you know, then very quickly, really quite amazingly quickly, there were clinical trials and of 3 different gene targeting therapeutics in this space, and a quick succession of approvals. And now we're in this remarkable place where we're actually thinking about entirely new things, new doses and formulations of these original drugs, combination treatments, and new targets for this disease. So it really did move quite quickly overall. So I'm not going to go deeply into the biology of SMA, but just so you all know, you know, we did have a unique opportunity here. This is a single gene disorder. It's much more complicated in CMT with multiple genes, a loss of function disorder, a recessive disease, and very unique genetics where there is the disease gene that causes the disease, SMN1, and loss of function mutations on both alleles cause SMA, but every patient retains this sort of backup copy of the gene, what we call SMN2. That version of the gene is alternatively spliced, most of the time removing an exon and encoding a truncated protein that's rapidly degraded, but some of the time that exon is retained and full-length normal SMN protein is made. So, you know, this genetic scenario really gave us an opportunity to sort of tackle this disease therapeutically in different ways. One, you know, is to replace what's missing, SMN1, but the other was actually to target the pre-mRNAs arising from SMN2, encourage exon 7 retention and more SMN protein expression. And even from the human genetics, we understood this was likely to be successful. And the reason for that is that we know the disease is highly phenotypically variable in patients, but patients who have more copies of SMN2 have less severe disease, indicating that more SMN correlates with less severe disease. And remarkably, there are individuals out there who have 5 copies of SMN2 and don't develop SMA. So that really taught us from the human genetics that sufficient SMN2 expression could prevent SMA. So this is where we are now as a field. We have 3 therapeutics. We have a viral gene therapy on onacenogene avaparivec, which basically replaces what's missing. It's an AAV9 with a SMN1-like cDNA packaged in it. It's delivered IV once in children 2 and less. And, you know, theoretically could be a one and done type of therapy. You don't redose this drug. The other 2 drugs actually target the pre-mRNAs arising from SMN2. Nusinersen or Spinraza is an antisense oligonucleotide that binds the SMN2 pre-mRNA and sterically sits on it, preventing RNA binding proteins from removing exon 7, thus generating a full-length RNA. And then risdiplam is a small molecule oral drug that you take by mouth daily with wide biodistribution that similarly increases exon 7 inclusion and full-length SMN protein expression. So how did the SMA field kind of get here? I know others in this room are very involved in this field and may have their own perspectives on this, but I think that there were some key elements that definitely helped us. Obviously, the single gene with these 2 versions of the gene was very helpful, but I think the field was very careful over the years in gathering the key stakeholders together, and during those meetings, really trying to address what are the key questions limiting therapeutics development. I really felt that during our annual Cure SMA meeting, I left that meeting each year knowing this is the key hurdle we have to overcome in order to move the field forward. And I think the field really reevaluated that quite frequently. Obviously, there are always sort of these fundamental questions, what are you going to target? How are you going to target it? Up, down, how much are you going to change it? Where is the therapeutic got to go? When does it have to go? And how are you going to measure it? In the SMA field, I think we were fortunate. We did have some key reagents that really enabled the field. We had human cell lines. We had some really good mouse models. But an important step that was made was that these were really well validated. And consensus was reached, for example, about how to do preclinical therapeutic studies in the mice, which mice, what outcome measures, what was the standard for the field. There was an investment in making these models available to everyone. So the SMA Foundation made a point of making sure that working with Arthur Burgess, for example, to deposit the lines at Jackson Labs, so they were available to everybody. And similar efforts were done with cell lines at Coriell. There was also thoughtfulness about what reagents were missing from the field. What were key things that the field needed in order to keep moving forward? And for example, a specific ELISA was made to measure SMN levels, and particular new mouse models were generated in order to answer key questions about cell contribution to the disease. I think, obviously, developing patient outcome measures that are relevant to the disease is also absolutely fundamental to success. And in the case of SMA, that turned out to be measures that were specific for infantile disease versus adult disease or older children, disease for older children. And in the end, clinical trials were done in parallel in those two groups of patients. And that was also very important to success, to make sure that the trials were appropriately designed. And of course, there was also, this effort to really recruit new people to the field. Obviously, none of us can know everything. We need to bring in experts who can help us with key questions. So in terms of, you always hope for these sort of key essential proof of concepts, these fundamental questions being answered to help you gain confidence that your therapeutic is going to be, useful in a human being. And I think, again, in the SMA world, the field was able to show fairly early that the disease severity very clearly correlated inversely with SMN expression level. And that was the case both in humans and in preclinical models. And at least for the most part, too much SMN seemed to be pretty well tolerated. So that really gave us a therapeutic window that was helpful. If you have too tight of a therapeutic window, it's going to make it really challenging once you get to the clinic. But it was clear in SMA that we had a pretty wide therapeutic window. We were able to establish that motor neurons were really the key cell type driving the disease, and that cell had to be targeted for an effective therapy. And that, even after disease onset in the mice, that you could reverse human relevant disease pathology. So this is really key, because you must establish in your disease models things that are actually going to be relevant to a human being. We further showed that there was an important window where SMA motor neurons were dysfunctional prior to them being lost. And part of that dysfunction was driven by a problem in the original development of the motor neurons. And this really emphasized the importance of getting in really early. And in fact, the clinical trials, when they ended up being designed, were, again, trials in which infantile patients were treated as one trial, but then separately older patients were treated. And many people predicted that we would never be able to have clinical effect in the most severely affected infantile patients. But of course, it turned out that those were the individuals who actually responded most robustly. Okay, so has it all been success? No, it hasn't, actually. You don't hear much about this, but there were definitely early failures in the field. We in the field, including me, thought that maybe just transcriptionally activating the SMN2 gene might be a good way to go. And we tried things like histone deacetylase inhibitors, and we looked at them in cells and in mice, and we could see that we could get an increase in SMN, we could get an improvement in disease phenotype in the mice. But then when we went to clinical trials with drugs that were already FDA approved, like valproic acid, really no benefit in patients. And in fact, no increase in SMN either. And so that really represented kind of a failure. And then there was an effort by Cure SMA to fund an unbiased small molecule screen and a cell-based assay, and a new chemical compound series was identified. It seemed to also transcriptionally activate the gene. Mechanism of action was worked out in academic labs. It went to a small company called Repligen and eventually to Pfizer. But again, it really seemed to lack sufficient potency and specificity. So these really didn't end up being successful, though ironically, I think there's some interest in returning to this idea of transcriptional activation in light of our more recent successes. So it really took these new gene targeting platforms to really gain success in the field. And, you know, I think it's really important to remember that each of these really started from academic and or nonprofit foundation efforts. So nusinersen or Spinraza, the splice switching ASO, that really started from an academic collaboration between Adrian Craner at Cold Spring Harbor and Frank Bennett and Ionis, and Chris has worked on these sorts of technologies very much as well. But, you know, they really it did really beautiful fundamental studies showing that you could identify very specific ASOs that would modify splicing of SMN2. And then, of course, early clinical trials were done and showed some efficacy. And then Biogen, of course, acquired the program and now sells the drug. But you know, this was really. I have these arrows here. Sorry, I'm terrible at this. That go back and forth. And I do that very intentionally because normally we think of drug development as sort of passing it on to the next stakeholder, and the prior stakeholder has no further involvement. But I think in this case, there was very continued active collaboration, even as the drug went from academia to small biotech to pharma, and that was part of the success. So omnisemgene abapiravec or Zolgensma, this similarly started from an academic effort. Brian Casper at Nationwide Children's, you know, developed the drug, showed efficacy, really remarkable efficacy in mice. He pulled in Jerry Mendel, who was a clinical trialist who really understood how to do gene therapy clinical trials. And then Brian started this company, Avexis, which was ultimately acquired by Novartis. So again, you know, an effort that really started in the academic space, but grew with robust collaboration. And then finally, risdiplam or Evrysdi, the small molecule splicing drug. This one started from funding from a nonprofit foundation in the SMA space, SMA Foundation. They recognized the potential power of small molecules that could change splicing of SMN2. They partnered initially with PTC Therapeutics, a biotech company, identified a series of compounds, and then that was acquired by Roche. But again, interestingly, this was not a pass-off. This was an example where very robust continued integrated collaborations continued, and in fact, ownership was maintained by all three stakeholders to this day, and profits that the SMA Foundation now reap are actually being reinvested in the field. So that's fantastic, I think. Okay, so, you know, I think it's easy to think, most of us, when we don't have a drug yet, and certainly I felt this way in the SMA space, is you just want to get a drug, and that's the goal. But I think what we realize in the SMA space is, yes, huge success, 3 drugs, but in some ways, it's kind of just the beginning, and it's surprising. So we generally think of this as a very linear path with an end. But I think the reality of this is that it's very iterative. And even once you're in the clinic, particularly in neurological diseases, I think, because of the special challenges there, you end up having to learn from your observations in the clinic and take those back to the lab. one of the challenges that we've seen in patients who are getting these drugs is that we're seeing a lot of variability in response. This is just a spaghetti plot from one of the trials with one of the drugs. I'm not going to tell you which, but you can just see when you look at the individual patients, there's a ton of variability in how they're responding. And it is very difficult for us to know what's happening here. We can't measure drug distribution in the CNS of patients. We can't measure the drug target in living patients. And it's very difficult to know what the evolution of disease pathology is, what we're able to change, what we're able not to change. So this makes it very challenging to figure out, how to best monitor and optimize drug treatments. And this just emphasizes the incredible importance, we all know it, but of novel biomarkers, as well as human biobanking, including autopsies. And that's a sensitive subject, but I really think it's so important. I'm struck in the CMT world how little actual pathology data we have about CMT. It's difficult because it is a slowly progressive disease, but I think that this is an area that we could really do more in. This is an example of some of the data we got from SMA after we made a really concerted effort to collect human tissues at autopsy. Each of these subjects represents an individual subject's spinal cord that we dissected at the time of autopsy. And this really teaches us that SMN levels in humans aren't static over the course of developmental time. They're actually falling quite precipitously between a fetal stage of development and postnatally. And obviously, these dynamics strongly influence how we think about targeting this protein. The other thing that we learned is that drug distribution is maybe not as we might think. This is, again, in patients who came to autopsy after being treated with nusinersen, and obviously biased to those who did less well. But I think what's important is that if you look at the distribution of the drug throughout the nervous system, the drug, of course, is not CNS penetrant, so it's delivered by lumbar intrathecal injection. You see these much higher levels of drug in the lumbar and thoracic spinal cord regions relative to the more rostral regions. So again, a really important practical issue that we've got to address. And this data I'm going to skip this, but this data, along with others, has really led to an effort to do trials with higher doses of nusinersen in patients. And those trials are ongoing, and also develop new generations of these ASOs that are more potent, may have better distribution, and may be able to be dosed less frequently. Okay, so where are we with CMT? That's what this whole meeting is about. But I came up with the same word. I think it was you, Clary, or maybe Edritz, who said momentum. I definitely feel like the momentum in the CMT space is really moving. And there was a feeling of like that in SMA too. Each year, there was a feeling that we had taken a step forward. And that we were making real progress. And so this is just two examples of this kind of progress. In terms of PMP22 knockdown therapies, there was this, oh, God, I'm terrible. I'm so sorry, guys. This paper a few years ago that was led from INS Pharmaceuticals, showing that an ASO knockdown strategy could work, at least in an animal model. But the challenge here was that particular drug didn't have the biodistribution in Schwann cells that we needed for human beings. And so this really led to the CMT RF thinking about this problem, investing in DTX, and now we know the great news about its acquisition from Novartis. Similarly, another recent example, the identification of SORD mutations by Stefan Zuckner's group, and the involvement in it resulting in increased sorbitol levels, applied therapeutics already having a drug that could potentially be efficacious, AT007, and now an ongoing clinical trial. So super exciting that these things are happening. I thought I just had to put in a quick plug about a target we're very interested in. This is for CMT2C and forms of distal SMA. Some years ago, we and two other groups described that dominant missense mutations of this protein, which is a cell surface expressed cation channel, can cause CMT2C and distal SMA. And they do so via a gain of channel function. So you can see here the mutants have increased calcium influx, whereas if you block the channel with an antagonist, you can completely block that excessive. Okay, I have 2 minutes. That's not so bad. We recently have built animal models and show that they have neuromuscular phenotypes, that they die early, they have relevant neuromuscular pathology. And if we treat these animals with a tool compound that blocks the TRPV4 channel, we can completely restore their survival in a dose-dependent manner and improve their motor function. And we're so pleased now to be working with Actio Biosciences, who are here and provided this data. They've independently confirmed that TRPV4 mutations do indeed lead to a gain of channel function, and they've surveyed basically all of the mutations, so they've done a really careful job, and have also showed that a new drug that they have discovered has really good potency against these mutant forms of the channel. And so we're really optimistic about potentially taking such a compound to the clinic. And so to prepare for that, we're doing a natural history study at Johns Hopkins of individuals with CMT. They do have phenotypes that are distinct often from typical CMT. And so we really think that a very careful, dedicated natural history effort is necessary. And I want to particularly highlight both Brett and Bapasha, who are here because they've been instrumental in this effort. And keys to success, I think very much in rare genetic disorders, the patient, families, and nonprofits play an absolutely vital role, and exceptional collaboration is absolutely fundamental. So thank you for listening, and I'm sorry if I went too long, but happy to answer any questions.
Any questions? I can bring the microphone.
Maybe I can go ahead and ask for Charlotte. So much for a beautiful presentation. So, and this might be a loaded question, and a big question is that you mentioned that with CMT, it's difficult and more complex because it involves so many genes. What is the right balance to take right now so that we can go fast, but also make sure that we're doing the right thing in terms of building more therapeutics that's really out there for CMT? What should be the right or the first take, I would say, beyond, the delivery challenge that we know of, and trying to get the therapies to the right target tissues?
Yeah, this is a real challenge for CMT, no doubt. And obviously some therapeutics that are generated are going to be very gene specific, and obviously there's a lot of interest in that given the recent successes. And I think that's appropriate to be investing in those gene specific therapeutic platforms. But I would also hope that in parallel, we're also thinking about therapeutics that may be relevant across the board for all CMT patients. So that's one thing I would say. But I do think some early real success would be really good for the field, because it'll get more and more people interested, more and more companies interested. They will see that it's feasible and jump in. I mean, I'm not in industry, but that's what I would hope. So I think, practically speaking, while I have, I take care of CMT patients every week, I don't want to leave anybody behind for sure. But I do think there's an important role to go on those targets that are most promising and most validated. I think it really is important to push those forward and focus efforts on those. If you're too dilute, you won't make headway on any one of them. So, it's a bit of a balance, but I think at this point, getting some success in the clinic with one of these would be huge for the field.
Hi, Charlotte. That was really fantastic. And thank you so much for being here today. We really appreciate it. When you look at, you're on the front lines, sort of treating patients with SMA. And when you look at, sort of the therapies that have been developed, you have a viral and antisense and then small molecule. How do you see that evolving? Is that sort of like a natural progression where, in terms of therapeutic development, or, is, one of those really, just going to serve as, sort of the end goal for the therapies in that space?
Yeah, it's a great question, Paul. So, I mean, I think as it went along, the timing of the approvals was not necessarily anything to do with the strength of the therapeutic, for example. And I think I thought that as new ones came, that they may become the preferred. But that has not happened, interestingly. And remarkably, and this is a whole nother set of biology we could talk about, they really have very similar efficacies. There is no data that I'm convinced of that demonstrates that any one of those three drugs is more efficacious than another. So, three are available, now in the US, we have newborn screening for SMA based on the observation that they're so much more effective when given very early. So newborns are being offered one of the three as an initial first line. Because gene therapy is not available at this point to children two or older individuals only get the choice of one of the others, two others. And it's true that most newly diagnosed families are choosing gene therapy as their first line, but it's rarely curative. And so most patients are ultimately going on to either risdiplam or nusinersen later in their disease. So no one of them seems to be immediately falling away. And I would say that all three platforms are equally exciting in terms of applicability to other diseases.
Well, thank you so much, Charlotte.
Yeah.
And before you get off the stage, one more, and then we really have to move on.
Sorry, but I'm just curious about the biology of SMN.
Yeah.
So I'm not in this field, but as I recall, it really wasn't known what it really does. I mean, what are some of the targets that it binds to, and how do you think it's involved in development?
Unfortunately, I just didn't have time to get into that at all. I could be up here all day if you wanted me to, but it's expressed in all cells. It has a very well worked out function in what's called SNRP biogenesis, small nuclear ribonuclear protein biogenesis. So it is important for the fidelity of bringing together SM proteins together with SN RNAs to form spliceosomal SNRPs. So the theory has been that it's alterations of splicing that actually cause SMA, but there's been lots of efforts to figure out what are those key alternative transcripts that actually lead to a motor neuron specific disease. And we don't know. We sort of skipped over the understanding of the biology of the disease and kind of lucked out with SMN replacement working. But whether it's that function of SMN that really drives SMA still remains in debate. It could be that there's alternative functions. It's a protein that in neurons has been shown to traffic up and down the axon, be present in the nerve terminal. So some have argued maybe it has a role in RNA processing specifically at the nerve terminal, and that's really how it affects a motor neuron specifically. But yeah, so functions have been clearly delineated, but the function key to SMA, yeah, that remains unknown.
All right. Well, thank you so much. And if you have more questions, we'll have breaks and lunch. Charlotte, before you get off the stage, I'm sorry, we can't leave just yet. So we have Susan Rudiger, who is co-founder and chief mission officer, and she has something special to say to you. Susan has been involved in the space since 2007, really, connecting so many different stakeholders to help fund research. And we are so lucky to have her at the CMT Research Foundation. So Susan, please go ahead.
Thank you, Edritz. So at the CMT Research Foundation, it's important to us to honor those who helped make us a success. And Charlotte, we decided to create a Pathfinder Award for Excellence to honor all of those who are serving the foundation. And when we were envisioning this honor, You were absolutely our very first choice. You, as Pat had said at the beginning, you were there with us from the very beginning. You said, you gave us your strategies, you gave us your successes, you showed us the paths that you took in the SMA field, and you challenged us so much to think differently. But then you said, and go find every company that's working in drug delivery. And it was that mandate that brought us to DTX. You said, go find them. And I worked with someone from our SAB and we found DTX and others. So thank you. You also helped us find our first CSO. You helped us guide our scientific strategy. You have been a friend. You have been a partner. You have been a mentor. And I'm so grateful for all your help. So Charlotte, we're lucky to have you. That to you is the first ever Pathfinder Award for Excellence in CMT Research. Thank you.